Because when we have this many people on a Zoom call, there's background noise. I'm guilty of it too. As you can see, I have a German Shepherd back there. I'll be on mute just in case he decides to bark at an inopportune time. So, just remember, and if you see me mute you, please don't be offended. I do it lovingly, because we want to hear everything that Anusha has to say. On that note, go ahead, Anusha, take it away.
Hello, everyone. Can you hear me, Kayla? Douglas, can you hear me? Yeah.
Yep. Yeah, loud
So,
and
Sound
clear,
great.
Anusha.
You sound great.
very good evening to all of you. So, I'm Dr. Anusha Pulavarty. So, I'll be sharing with you my research that I've been doing since 1 year. You might have seen me collecting samples from your lands. So, I'll be sharing some of the information or the findings that I have been observing or recording in the past few months. So if everything is okay, I can share my screen so that you can all see my presentation. Just a second. Yeah, can you see my screen?
Yep, you just got to start sharing the presentation. Yeah.
Yeah, can you see my presentation? Now I'll just put it
Yep.
in the presentation mode. Okay,
Yeah, perfect.
all good. So again, so the topic that I have been working on was investigating nematode biodiversity in Irish peatland habitats. So within a second, I'll be explaining what nematodes are and what are their role in ecosystem and everything. So my current project is funded by Irish Research Council and GRI. So Douglas is my enterprise partner, my mentor, and Dr. Thomai Kakuli Duarte is my mentor from the university where I work at. It's Southeast Technological University based at Carlow. So I think most of you are already aware of what are peatlands. So I just want to summarize a little bit, a few bits and pieces about peatlands. So peatlands or peat is composed of varied mixture of decomposed plant materials that is unique to various natural peatlands. And peatlands are also referred to as bogs, fens, mires, moors, or swamps and musks. And then coming to the importance of these peatlands, it is known that peatlands only constitute 3% of the total Earth surface, but the amount of carbon that they store, these unique systems, is almost 30% of world's soil carbon. And then the other most interesting fact about peatlands is that per unit area, the amount of carbon dioxide that the peatlands capture or sequester is at least 10 folds higher than the similar area of Amazon forests. So you can imagine how important these peatlands are for our ecosystem and for the planet. And so they also play a very important role as carbon sinks, and so they have a very important role to play in the global carbon cycle, etc. So as we all know that there are different layers of the peatlands. The topmost is the acidic bog peat, and then the black fen peat, which is a basic kind of pH. And underneath that would be a lake. So these are the different layers of a peatland. So it's just like an upside down of a sea. And then these peatlands are present throughout the world, and Eurasia, North America, Northern Asia, and many parts of Latin America as well, and Africa. So these peatlands are house for many unique for flora and fauna. So then coming to the importance of nematodes, why are we so much concerned about nematodes, and why have been working on investigating the nematodes that are present in these peatlands? So nematodes are microscopic in nature, as you can see in the pictures. They're unsegmented worms, and they're known to exist in every habitat that is known to mankind. And these creatures are the most abundant creatures on the planet. So it is being said that out of every 5 animals, 4 are nematodes. So that's how much abundant they are. So there are nearly 57 billion nematodes for every single living human being. So because of their highest abundance and because of their huge biomass that is constituted by the nematodes, they are most important organisms. So by studying nematodes, you can pretty much understand the ecosystem of that particular area. And then, so why are we using nematodes as bioindicators? As I mentioned, that these are the most abundant creatures. And if there are any changes in the environment, these nematodes quickly respond to the changes. And so we can easily detect the environmental disturbances, pollution, or can understand if there is an effect of heavy metals, or if there is any future harm that could be caused to the plants or animals or insects or etc. And then these nematodes, as you can see in the picture underneath, in the food web, they have their position is placed in different trophic levels. So by being fed by other creatures, or by being feeding upon the other creatures, they play a very, very important role in the food web. So there are different types of feeders in nematodes, like bacterial feeders, fungal feeders, plant feeders, omnivores, and predators. So based on their feeding habits, they are assigned to CP scales, which is called colonizer persister scales, which it starts from 1 to 5. So 1 would be the colonizers and 5 would be the persisters. So they act and they respond very quickly to the environmental changes. So because of these different unique characteristics of nematodes, by being placed on different parts of the food web, reacting to the changes, they are the best indicators. So they can be used as bioindicators to understand the pollution or to understand the restoration in my case.
So if I could ask before you move on there, Anusha. So
Yeah.
colonizer persister. So the what's the scale there? So the colonizers are just for the disturbed habitats, and the persisters are for the mature
More stable.
habitats that
Yes, that's
haven't
correct.
been interfered. So woodlands or intact peatlands, for example, would be
Yes, we'll
typical
have more of
habitats
them.
you would be finding these in. Yeah.
Yes,
Okay.
that's correct. Yeah. So I just forgot to mention, like, if I'm talking too much of a nonsense, or if something is not
No,
clear,
just
you can.
excellent. Just
Yeah. So
slow down. That's it. Take your time. Take your time. Is
yeah.
very good. Thank you.
So please do not hesitate to interrupt me as and when required.
Yeah.
And
Excellent.
proceeding. So
Thank you.
just to say in short, it said that if all the matter in the universe except the nematodes are swept away, our world would still be dimly recognizable. So as disembodied spirits, we could still investigate it and find it's like, if we can find out if there are mountains, hills, or rivers, or lakes, or oceans that are present just by studying the nematodes. So that's how important nematodes are in the environment.
So basically, you could reconstruct what
You can
ecosystem was there based
reconstruct.
on the
Yes.
nematodes that you would
Yeah.
find. Yeah.
Yeah. So if I'm allowed to go to the Mars and do some sampling, I could pretty much tell, like, what's there in the Mars, even without sending all those expensive satellites.
Very good. So if you take away our bogs that you're investigating, you'll be able to tell us what type of bog was there. That's great.
Yes. Yes. And now proceeding. So what is my project concerned about, and what am I doing using nematodes as bioindicators? So this is to evaluate the restoration efforts that are being done by Green Restoration Ireland, to see how well these bogs have been restored, or how well they are similar to the healthy bogs or to the rich fen kind of peats. And so this can help us to completely evaluate the soil health status of a particular site or the peatland habitat that we are investigating in. So that's what I'm trying to find. So the overall work that I have been doing is like trying to take the peatland samples from different peatland habitats and then bringing them into the lab in a controlled temperature kind of bags, like insulated, thermal insulated bags. And then once the samples are brought into the lab, we try to very nicely mix them and homogenize them. And then I was dividing each sample into two halves. One half I've been using for the morphological identification. So in a minute, I'll be explaining what morphological identification means. And the second half I've been using for the molecular analysis, or the identification of nematodes using DNA or molecular techniques. So once the morphological and molecular identification is done, and once I get to know what are the nematodes that are present in particular peatland habitat, I feed in all this information into an online tool, which is called Ninja, that stands for Nematode Indicator Joint Analysis. So it's an online tool which can does the calculation based on 40 different nematode based matrices, and then it gives us an indication about what's the ecological status of a particular peatland habitat. So that's what my overall research work is all about in relation to
Mm.
nematodes and peatlands.
So this
So
is this is microscopic work, Anusha, yeah? So under the
yes, it is.
microscope.
Yeah, the
So
morphological
yeah,
work is all microscopic, and the molecular is mostly like gene oriented. Yeah, so I'm studying a
yes,
particular genes and then identifying nematodes based on those presence and absence of the genes.
yeah. And just quickly, if I can ask, so in
Mhm.
terms of, because we were collecting pretty substantial soil samples, so how many? You don't go through every bit of soil or peat when you're looking under
Okay.
the microscope. You take a
Yeah.
sample of that, do you?
Yes. So for example, if we imagine a habitat to be in a square shaped, for example, so I try to collect 5 representative samples of that particular land in a W manner. So I take it like if you imagine a W on a square, so it's like
Mhm.
towards each corner, 4 ends, and one towards the center. So in that
Yeah,
way, I take 3 Ws. So it's like so one W represents one composite sample, so it means
yeah,
that it's an indication of the whole site. And the second W would be a repetition of the next. It's like a next replicate. So the third one would be a composite replication of the third replicate. So I try to take 3 Ws in one site.
mm,
So it's pretty much has a complete representation of that particular site. So that's how
yes,
I
yeah,
keep doing. So every habitat or every site that I do, I take 3 composite replicates. Yeah,
yeah,
and
yeah.
then
Very comprehensive.
each replicate, and then it's divided into 2 equal halves. One would go for the molecular work and the other one would be for the morphological work.
Mm.
That's how it's being
And
done.
but I suppose so that's exactly so that's what we were doing in the field in terms
Yeah.
of in on the peatland and the grasslands and so on. But then when you brought that back to the lab, so I wasn't there for that bit. Um, how do you so you have a chunk of peat basically
Yes.
for doing the plating it up and looking at under the microscope?
Yeah, so
You don't
once
do
I
every
have
bit of peat. Do you take a sample out of that peat just as a question?
Yeah. So once I have, for example, if I have one W, so what that's one replicate of one habitat. So one replicate, I just homogenize it very well manually, and then I divide for example, 500 grams of soil into 2 equal halves. So the 250 grams of soil that is used for the morphological analysis, I try to use some literature survey to see how the nematodes are extracted from this peat soils. But out of my maybe it's a lucky thing or an unlucky, unfortunate thing that there were not much literature available on peatlands. So I had to optimize a particular protocol for extracting the live nematodes out of the soils.
Yeah,
So I
which you
optimized a
published.
protocol for
Yeah.
it. Yeah, yeah, which was published. So I can send you the links for anyone to refer in future. So I optimized a particular protocol so that we can extract the nematodes can crawl out of the soil samples, and that
Mm,
nematodes are then they undergo a little process and they're fixed onto the glass slides, which are then observed under the microscope for morphological identification. So that's the first
mm,
half. And the second half story is like I try to isolate DNA from those soils. And that, so I do two different molecular techniques. One for sequencing of the DNA, which is being outsourced. And then once the sequence of the DNA is obtained, I try to analyze what are the nematodes present in it. And the other thing is like another fingerprint technique, which I'll be talking through when I'll be proceeding in my presentation.
mm.
So that's another technique that I do for molecular identification of nematodes. Okay.
Thank you.
Any other questions, Doug, or shall I proceed?
No, no, that's perfect. No, no, that's it. My curiosity has been satisfied. Thank you.
So we're proceeding to the next one.
Hopefully, it
So
was
I'm
relevant for the people listening as well. So,
Okay, so I'll be representing the first 3 sites that I've been doing. The first site, I think, if Dhoni is in the audiences, then that's Dhoni's site, and the second one is Cody's site, I
mhm.
think, and then the
Yep.
third one, I do not know the name of the
This
person.
is Haslam's. So shall I
Okay.
just maybe give an overview of the habitats, Anusha? So
Yes. Yes.
the first slide there you can see. So in the middle you have the raised bog. So this is essentially semi-intact. It's typical of a raised bog in the Midlands. Is about 25% of the original bog left. The rest is cut over or reclaimed pasture. So this is kind of semi-degraded raised bog in the middle, and then you collected towards the edge where it's more
Mhm,
degraded.
mhm,
We also looked at the peat pasture, which of course is the focus of our EIP. And what we were looking for here was bioindicators of peat grasslands. And then we were also looking at the cutover So there was typically when the bog is drained, trees and scrub come in. So we're looking at where the cut over scrub and woodland and where it was re-wetted and non-re-wetted just to see if we could see any difference. In the middle, so we have two fen sites. So the one in the middle, the site two. So this is Listaff fen. So this is a listed SAC. So we have the fen itself, and then we have the reclaimed peat pasture. And then the third site was so we actually had trouble finding peat there. There's a lot of lime. So we also did another site to get a because the just to explain for the audience,
Mhm.
when peat is reclaimed, typically a bog, a raised bog is an ancient lake that's essentially filled up with peat and keeps raising up above the water table. So typically, because we're interested in the peat grassland, when the peat has been reclaimed, typically it's down to the fen peat. So we wanted to look at the original habitat just to see if there was any similarity there. So in this case, the third site is Island Fen SAC. So this is an intact fen site. So there we go.
Okay, thank you, Doug. So the next, so when it comes to the morphological identification, what I do, like as I explained to you just now, like once I have the soil, I try to do a very simple kind of protocol that I have been optimizing in the lab, and I allow the nematodes to crawl out from the soils into the clean water that is kept underneath the soil samples. So once the nematodes are all collected in the water that is kept underneath, I try to fish them out. It's literally fishing them out from the observing under the microscope, and they fish them out from the water. And once they are all collected into a small glass blocks, and once they're collected, we try to do some kind of we kill them using formaldehyde so that they can be permanently mounted onto the glass slides and they can be observed and the pictures can be taken under the microscope. So when I say like if a nematode is feeding on a bacteria, it's called a bacteriovore, and the one that's feeding on the fungus, it's fungivore, and the one that feeds on the plants, it's herbivore, and then if it's feeding on insects or other nematodes, it's a carnivore and omnivores. So as you can see, the top picture, it's the illustrations that were obtained from the literature. And these pictures that are underneath are the microscopically captured images under the microscope by us. So you can see that the mouth part of the bacteriovore it's white so that it can suck in the bacteria. So it has a different mouth part compared to that of a fungivore, which is very sharp kind of feature that it can pierce through the fungal spores and fungal hyphae. And then the herbivore, it has a different kind of characteristics. Like it holds a stylet, which you can see, like it looks like a needle, and the back and forth movement of the stylet or the needle is guided. Can you see my cursor on the screen, Doug? Yeah.
Yeah, it's moving around a bit, but yeah,
Yeah.
there's a black arrow. Yes, yeah.
Yeah. So the herbivore, it
So you're
has
looking
this.
at the photograph below, yeah, yeah.
Yeah. So it has a needle-like shape so that it can pierce the plant roots, and the movement of this needle is guided by two little knobs underneath, which are called as stylet knobs. And then the carnivore, it has a tooth kind of characteristic, which you can see in the picture. So it's like a tooth for the nematode. And then the omnivore again has a spear, which is very sharp, and it helps in piercing the organisms and feeding on them. So these are the mouth parts that are distinct and very much specific for different feeders.
Mm. So
Anusha,
just,
we have
I
we have
think,
a question in chat. What scale
yeah,
are these nematode pictures at, by the way?
These nematode pictures are at 100x, so it's like 100,000x magnification,
hundred
and they are
thousand times magnification, yeah.
thousand. It's
Oh,
thousand
thousand,
times
a thousand,
magnification. Yeah,
yeah. Okay.
is the total magnification. So yeah.
And just to clarify, Anusha, so the herbivore essentially the stylet is for piercing roots. Is
Yes.
that what the herbivores are feeding on? Yeah.
Yes, it starts piercing the roots and it enters into the root system. And once it enters into the root, it tries to hijack the a particular cells or the root cells of the plant, and then it establishes a feeding site, and then it tries to feed slowly onto the plants. And once it establishes the feeding site, it keeps continuing the feeding process, and then eventually it kills the plant. So these herbivores are very, very potentially economically potential threats for the growth of these plants. So there are different herbivores which I'll be presenting eventually. So it was a surprise for me that peatlands had many of these herbivores, which could be a threat for crops and paludiculture. If as paludiculture is the future of restoration of the peats, so it's very important to note like what kind of herbivores are present in different habitats. If they are not controlled, they can cause a threat for the total agricultural fields and to the crop output.
Anusha, could you do me a favor real quick? At the bottom of that screen, can you click hide? We just want to be able to see the bottom of that picture. Sorry, it's sneaky. I forget to do that.
Sorry.
Oh, the
Oh, you're good. No
presentation is gone.
worries.
Oh,
There you go.
it's
Can you
back.
see that now? Yeah.
Very good.
Perfect.
Very good.
Thank you.
And
We just
and
didn't
just
want
a
to
very
cut
quick
off
question.
those photos because
Yeah.
they're so
Yeah,
impactful.
Yeah. And just one quick question in terms of infestation. So it's not one nematode that will kill a plant, Anusha. It would be hundreds or thousands. How many would be attacking a typical plant?
So it all depends. So some of them are like, okay, once they establish the feeding site, they try to reproduce within the plant. So because the plant is their host, they keep feeding and then they reproduce inside the plant host. So some of them produce cysts. So they are called as potato cyst nematodes. They cause cysts like the ones, like the tumors that are caused in human systems. So these nematodes cause cysts in the plant roots. So these cysts, they are nothing but they are the female bulged vulval region. So it depends on it essentially requires if they are cyst nematodes, even if one cyst is present, one cyst could carry at least thousands of females. So the potato cyst nematodes are quarantine pests for Ireland. So those are the problem of that. And then there is another pest which is called Bursiphyllingus, which causes infection in the pine trees. So again, the threshold for those pine trees is like if there is single juvenile present. So if there is a single juvenile present in the soil, then it can ruin the forests. So it's enough to establish the feeding site and to reproduce, and it keeps going. So that's again a threat. So that's also a quarantine pest in Europe and in Ireland. And the other one which infects the tomato plants, it's called as a Melodogyne species. So for that, it's if I remember correctly, I think it's 20 females. Yeah, if there are 20 females existing in the soil, that's the threshold limit. If it exceeds that, then it can destroy the crop completely. And they are potential threats. They cause huge crop losses worldwide. Yeah.
Right. Very interesting. Thank you.
Shall I proceed? So moving on to the features of the plant parasite, as I mentioned to you, this is the feeding needle, which is guided by this little knobs underneath. So the back and forth movement of this needle is guided by the knobs, and it starts feeding onto the plant roots. It establishes a feeding site and keeps feeding on the plant and kills the plant eventually. And they are the reason for thousands and thousands of billion-dollar crop losses worldwide. And and as you can see in the pictures, the first three pictures are the mouth parts, and the one that is on the right side, it's the tail, and then you can see the vulval region of the female nematode. And then moving on. So for the morphological identification, as I mentioned, once we collect the samples from different sites, we take the pictures of the mouth part, the tail shapes, and then these nematodes also have many special features like these are called as the spiral and different shape amphids. They are like the sensory organs for the nematodes. So based on their shape of these amphids, tail, mouth, and many other features like digestive parts or the bulbs that are used for the digestion of the food that the nematode ingests. So based on these characteristics, we try to assign families to the nematodes. So once we assign the families, we try to use some Miro boards that can help us in getting a visual fingerprint of a site. So for example, this is one picture, the different colors that you see on the right side underneath. This is a visual fingerprint of one habitat. So it starts on the lime-colored images. The lime-colored ones represent the nematodes that feed on the fungi, the fungivores. And then the green colors is the color indication for the plant herbivores that feed on the plants. And the blue to purple, more or less, are the bacterial feeders. And then the yellow ones are the omnivores and the orange ones are carnivores. And always in every site, we have some nematodes which is very difficult to identify. So I just have one or two of them, which is unidentified kind of nematodes. So in this, as soon as we look at this Miro board of one site, it can just give us an indication like how many fungal feeders, how many bacterial and omnivores and herbivores are present in one particular site. So roughly for each site, I try to take at least 70 to 100 nematodes, but collecting them, fishing them out, it's a very tedious process. And fixing them onto the sites and taking the images, it really requires a lot of expertise and patience. So that's how we have the characterization based on the morphological features of the nematodes. And then coming on to the molecular work. So what I do is like the other half of the peat soil samples that I collect from the field, I try to extract the total DNA from those peat soils. So these images that you see on the slide are the quality of the DNA. So we try to check once the DNA is isolated, we try to see the quality of the DNA and the quantity of the DNA. To quantify the DNA, we use a method which is like very much similar to a spectrophotometric method. So we'll definitely have a amount of DNA that's present. It's represented in terms of nanograms, so per microliter. And to check the quality, we try to do some gel images. So all this, that the bands that you see are the DNA bands that are observed under the UV light. So it's a quality check of the DNA that is extracted from each site. So each line represents a DNA amount of DNA that is extracted from one peatland habitat. So once the DNA is extracted, this is sent out to the sequencing company to sequence the whole, the particular region of the DNA that is very specific to the nematodes. So based on that, we try to identify what kind of nematodes or nematode families or species are present in one particular habitat. And then the
And you mentioned,
yeah.
Anusha, just to come back. So you're saying this is a quality check. So
Yes.
just looking at each of these bands, are these good? They're bad? Could you just maybe indicate if they're good or?
Okay, to just show like to say like whatever bands you're looking at right now, they are more like very intact and very good quality DNA. So for example, if you want to say a band that would look, which indicates a bad quality of DNA, it would be like it would be very sheared. For example, in the second image on line 6, the lane 6 that you see. So this is still a good amount, but if the shear keeps continuing and if it doesn't give a bright band, that means it's a very low quality DNA. So if we send out that kind of low quality DNA, it's very difficult for the sequencing machine to sequence it efficiently. Yeah. So we try to keep the quality as good as possible. So even for this kind of techniques, it requires a very aseptic conditions, not to contaminate and not to reduce to keep a good quality DNA. It requires very molecular techniques that is very safe for the lab environment, and try to use masks and gloves, such kind of laboratory safety equipments. Yeah.
And just very quickly, so if the DNA was maybe more spread than it is in lane 6 and 7, you'd need to repeat the
it's
sample
still good.
collection.
It's still good. Yeah. So we need to repeat the sample extraction again. So we need to get the fresh samples in and then repeat it again. Yeah.
Okay.
And then proceeding to the next molecular technique that I do. So the first part of DNA, so I try to extract DNA in four different replications. So the 3 replications are sent out for sequencing company, and then the next replications I try to perform a different molecular technique, which is called as RAPD, which stands for Random Amplified Polymorphic DNA. So in this image, I have put 2 gel images. So each gel image represents one habitat. So to simplify what this means is like I try to amplify different sections of DNA that is extracted from a peatland habitat, and then the different sections are then amplified using specific primers that are being picked up from the literature, and then as you can see in the image, there are the band patterns that are formed. So each peatland habitat would have a different banding pattern. So based on the number of bands that are observed in all the lanes, I try to manually record them. And then once I have the number of bands, Then I try to understand what would be the molecular weight of each band. So to get the reference molecular weight, the first lane has a marker lane or a ladder lane, which represents different molecular weights. So if a band represents at this is obtained at this level, then it would say like it's a 500 base pairs would be the molecular weight of it. So that's how I try to take the molecular weight of all the bands. And then once I have that data, then I try to do a dendrogram, which kind of tells us how similar are the peatland habitats to each other and how they are closely related or distantly related. So this is another kind of fingerprint technique which can help us in visual fingerprint of each peatland habitat. So by doing so, we can try to understand how the restoration has happened, how a particular habitat is close to a healthy bog, or if it's close to a degraded kind of bog or to a wasted peat, etc. So for example, if you see this image, the dendrogram shows us that the first 4 habitats, the IFPG, NRW, WP, IFPG, RW, RGI. Okay, so all these are grasslands. So these grasslands, and again, it kind of made sense because they were all grasslands, and the nematodes that were present in the grasslands were mostly the parasites that feed on the grass. So based on the ecological status of these lands and the nematodes that are present in them, they were all stacked together. And then the woodland habitats were stacked together. So W stands for woodland habitats. And then the FP stands for the fen peats. So again, based on the organic matter and the nematodes present in them, they were stacked together. And then the DBH, DBL, HPL, HPH, these are all the raised bogs. So they were all again stacked together in terms of the nematode communities. And then the cutover scrubs were far apart. So apart from them, the others were kind of made sense in terms of the organic matter and the nematodes present and the vegetation growing in those habitats. And do we have any questions here, Doug?
No, that was very well explained. Thank you.
And moving on, so the first part of DNA that was extracted, that was sent for the sequencing. So the sequencing company sequences the DNA and then it gives us a data which part of it looks like this, which says that what are the different nematode families present in different habitats. So the x-axis represents the different peatland habitats, rough grazing, cutover scrubs, woodlands, improved fen peat grasslands, fen peats and rich fen peat, and then the bogs and the wasted peat. So in each habitat, what are the families? So the color codes that are present here, it represents that if it is in a blue color, that means that the amount of these nematode families are lower. And if it's a red coloration, that means the nematode family is very abundant in that particular habitat. So, and there were a total of 23 nematode families identified, among which 9 of them were plant feeders, herbivores, 7 were bacterial feeders, 2 were insect parasites, and 2 fungivores, 1 algal feeder, and 2 predators. And the ones that I've highlighted in the red circles are all the plant feeding nematodes, which are known to cause huge crop losses worldwide. They're all herbivores. And so again, the same data is being represented in a different form, where I try to understand the composition of overall nematode assemblage in different peatland habitats. So the red ones are all the bacterial feeders. So irrespective of the peatland habitat, they were all dominated by bacterial feeders. And the next dominating feeding group was plant feeders, which are herbivores, followed by fungivores and some omnivores and algal feeders as well. So the bar graphs underneath, it shows that the most of the bog habitats or the healthy form of peatlands were dominated by bacteriovores, whereas in case of plant feeding nematodes or herbivores, in the those percentage in the bogs was very low compared to other kind of habitats. So it's just the other way, in an inversely proportion. So the bacteriovores were higher in the bogs compared to that of the grasslands and the scrubs and the woodlands, whereas the plant feeding nematodes were higher in the woodlands and cutover scrubs compared to that of the bogs, healthy bogs or raised bogs and the fen peat. So,
And and then the plant feeding nematodes, Anusha,
mhm.
that's for plants. It wouldn't include mosses.
It would include
That
mosses as well. So
okay.
the usually the plant feeding like algal feeding nematodes and the plant feeding nematodes would all be included in the mosses. Would all be included in the herbivores. Yes.
hmm hmm No, it's just because again, the healthy bog would be 80% plus moss and a lot
Mm-hmm.
less vegetation than on the cutover. So maybe that was a factor in
Reason.
the dominance of the bacteriivores.
Mm. Okay. And then I would like to highlight again that, yeah, the blue colored bars. So I try to represent the different nematode families that were obtained from the molecular analysis and try to see what is the dominant kind of species in different habitats. So this is just another way of representing the same data. So just out of my surprise, because I identified so many plant feeding nematodes, I try to report them separately as a different publication, which is my very recent publication. So I'll be talking also about one particular highlight in course of time. So okay. So once I have
And
all this information of nematodes from the molecular identification and morphological work, what do I do with it? I try to do an analysis, which I mentioned earlier, which is called as NINJA. It stands for Nematode Indicator Joint Analysis. So, what does this analysis tell us? So, it takes in the information that I feed, observing based on my investigation, and it uses approximately 40 different nematode based indicators or indices or metrics and tries to do calculations and tells us, divides the different habitats into one of these four quadrants. So the most of our peatland habitats fell into these two lower quadrants. The left one represents, if the peatland habitat comes under this quadrant, then it is said that that particular habitat is very degraded. It's a depleted environment. It has very high carbon to nitrogen ratio, and it is dominated by fungal feeders, and it's a very conducive environment. But
Mhm,
most of our peatland habitats were falling into the right lower quadrant, which means that the particular environment is very much matured. It's a fertile environment and it's a moderate carbon to nitrogen ratio, and it has a bacterial or fungal ratios, and then it's a very suppressive environment. The suppressive kind of environment, it means that this particular habitats would have low, they would suppress the growth of opportunists or the bacterial feeding nematodes, and it has a more stable kind of environment compared to the one which is conducive, which means it's a degraded or a very much disturbed kind of environment.
mhm,
So this results is based on only the 14 habitats that I'm representing. I'm still waiting on results from the sequencing company for more habitats. So once we have those results, I can have a more mm, how do I say, it'll be more conclusive kind of results. But as of now, I could see that most of them fall into this quadrant, and the wasted peat, rough grazing, and woodland revetted falls into this kind of quadrant, which means it's a degraded, depleted, had very much had a lot of human intervention, and it's a disturbed kind of environment. So
mhm.
that's what I can say as part of my work till now.
Very good.
And then
And then
yeah,
I think
go
in
ahead.
the next slide, yeah, this is the next slide. So somebody was asking, Jack, I think was asking, are the nematodes, the plant feeders, are they species specific or are they general? But I think that's what this slide will
Yeah,
you'll
it
be
is.
explaining that in this slide, I think.
Yeah, Roger. So this is the most important finding that I have observed so far in the peatlands. So as I mentioned, I've noted a lot of plant feeding nematodes. So it is very much species specific. It's very much host specific. So irrespective of the peatland habitat, all of them had this nematode, the plant parasitic nematode belonging to the family Tylenchidae, which usually parasitizes mosses and heathers. So as we or as just now Doug mentioned that most of the peatland habitats have some moss and trees as well. So this was commonly found. So PPN stands for plant parasitic nematode, PPN family that was present across the peatland habitats.
Yeah.
And then the next one was Hoplolimidae, which kind of feeds upon the gorse plants. And they were present very high in the rich fen peat and in the cutover scrubs and in the improved fen peat grassland as well. And then the other economically important PPN belongs to the family Pratylincidae. These are the nematodes that feed on the grass. So and this was very evident in all the grasslands. And this is a nematode which is currently categorized as a quarantine pest or upcoming threat for Ireland.
Mhm.
And then the other nematode family is Heteroderidae, which kind of feeds on the grasses, nettle, and oat grass and sedges. So they were found in these habitats, which is rough grazing and improved fen-peat grassland, rewetted and non-rewetted. And the Melidogynidae is again another quarantine pest for Ireland, and it should be controlled because it is very economically important, and it kind of infects most of the crop plants, and it kills the plants eventually and reduces the economic outputs. So if this is not controlled, then it might even affect the other agricultural fields. And if we are introducing paludiculture, because mostly paludiculture would involve crops, so there is a threat for those crops to be infected by Melidogynidae nematodes.
So
And
when you say most crops, Anusha, so it
Mhm.
can be anything from what cabbages to wheat or.
Cabbages, yeah, it can infect cabbages, potatoes, tomatoes, and then berries, and it also infects carrots, and it can also infect plums, many fruit varieties, and yeah, so it has a very wide range of host range. Yeah, and
So it's
the
a real generalist then. It's not a.
Yes, yes, yes. And it has a capability to, I don't know how, but it tries to adapt itself to different crops. So it has a very wide host range, and it is known to infect many crop varieties. Yeah. And then the other most important nematode which was found in these fields was belonging to the family Criconematidae, which was mostly present in the woodlands because they infect oak, birch, and holly. So why I'm representing this data is because this data is still unknown in Ireland, and if these nematodes are not controlled, they can be a potential threat for the paludiculture crops and also to the agricultural crops that are currently growing in these peatland habitats. So these needs to be controlled, or wisely we need to select crops that will not be potential hosts for these nematodes. So this is one of the most important finding, which I have already published in my recent publication.
Mhm.
So
So
which
just
can
before you
be.
move on,
Mhm,
quickly, if you can go back
mhm,
1 second. So just so again, the first one, the Telenchidase, so sphagnum and heathers. Again,
mhm,
healthy bog would be 80% plus moss, plus heathers, and certainly reindeer moss as well. Gorse plants is also there a bit in the grassland. So maybe
mhm,
also, so gorse being a legume, a shrub that typically invades dry peat cut over. So maybe it's parasitizing some legumes in the meadows, in that case. And then the last one, you were talking about crops. So oak, birch, holly. So again, one of the other things would be paludiculture forestry. So holly dies off when you re-wet cut over woodland, but birch would be a possible crop. Yeah.
okay.
And real
And
quick, when you say controlled, someone had asked in chat,
Oh yeah.
what do you mean by that? Is that controlled through planting certain plant varieties, or is it through like
yes.
pesticides? So just expand on that a little bit, please.
Okay, so control could be of anything. The first thing would be to select some resistant varieties. So if we are selecting some resistant host plants that cannot serve as host for these pests, then it could be one way of controlling. The other way of controlling would be to introduce certain kind of techniques like biofumigation, or using certain kind of plants that have the potential to fumigate the fields. So it releases some kind of compounds that can kill these pests. So that could be
Mm.
another approach. Pesticide would be the very last approach, which would never be recommended by organic farmers, because this can ruin, as we all know, like they have their own chemical side effects. So that's not recommended. But first would be to select resistant varieties, and the second one would be to use some biofumigation crops that can, like for example, um, mm, okay, the products that are organically obtained from the biofumigation that have the potential to fumigate the affected fields. So those two could be the immediate approaches to control the spread of these pests or nematodes. Yeah.
And can you actually use other nematodes to control the nematodes? I mean,
Yes,
that's
that's
sold as
that's
control.
Yeah, so that's another way of. Thanks for reminding that, Doug. Yes, that's another way of controlling the spread of these pests. So there are other kind of beneficial nematodes, which are called as which kind of parasitize the harmful nematodes or other harmful insects. So they can also be used for controlling the spread of this infection or the spread of these pests. So that's another way of controlling the spread of plant parasitic nematodes. Yes.
Mhm.
Or selecting the resistant varieties or the plants that cannot be host for these nematodes. So even if we can use that kind of approaches for a while, then these pests can be controlled, and then it will stop the creation of the next generation of these pests. So in that way, we can stop the spread of these pests.
Mhm.
Okay, and another important thing
Okay.
that I forgot to mention is like the green circles that I've highlighted. I've seen that only in case of improved fen peat grassland, the rewetted portion of this particular habitat had lesser amount of these pests compared to the non-rewetted. So it's very difficult for me to say at this stage if this is a very significant observation, but it's something that could be of interest, which says that maybe rewetting is trying to control the spread of these pests in these particular habitats. So you can see that in the rewetted portions, the abundance of these pests are lower compared to the non-rewetted counterparts.
Mhm.
Yeah. So
Good.
this is the publication which is present online, and we can use this link to see the detailed information of this finding. So summarizing or concluding the overall presentation. So in the presented dendrogram that I showed earlier on, it says that the grassland habitats are all clustered together, and the fen peat were clustered together because it's very much makes sense in terms of the organic matter and vegetation growing on in these habitats. And then the raised bog habitats were clustered together, and the woodlands were also again found to be closely related to each other. So it is based on my observation in terms of vegetation and the nematode communities that are present in these habitats. So that's one important finding that I observed. And then a total of 23 nematode families were identified in these peatland habitats, among which 9 were plant feeders, 7 bacterial feeders, 2 insect parasites, 2 fungivores, and 1 algal feeders, and 2 are predators. And then, as per the European Plant Protection Organization guidelines, it says that the current quarantine pests in Ireland are belonging to the family Apheloncoridae and Heteroderidae, and the emerging pest of concern is belonging to the family Anguinidae, which can infect the bulb species, and then the Melodogonidae, which I just mentioned, which has a very wide host range that infects the croplands. So in the studied peatlands, both the molecular and morphological results suggest us that the PPNs belonging to Anguinidae, Pratylincidae, Melodogonidae, and Heteroderidae were detected above the permitted threshold levels. So, which emphasizes the importance for the policymakers and farmers and the ecologists to consider and select potentially resistant crop varieties to stop the spread of these potential pests. And then the molecular data also suggests that the peatland habitats, most of them have a suppressive, fertile, moderate C/N ratio and bacterial dominant kind of matured environment, whereas the wasted peat, rough grazing, woodlands, rewetted have a degraded, depleted, and a conducive environment. So the overall outcome of the study would be very beneficial for the enterprise partner in evaluating the peatland restoration efforts and the solution. So once the restoration is efficiently done, it can very well help in reducing the greenhouse gas emissions and fight the climate change and restore the biodiversity and water quality. And the research also helps in contributing towards the UN sustainable goals, particularly in terms of climate action, partnership for goals, life on land, and improve the overall good health and well-being. So finally, I would like to thank all the peatland owners, farmers, and my enterprise partner, Doug, and my research team at SETU, and my mentor at SETU, Dr. Thomai. And thanks one and all, and to my funders, Green Restoration Ireland and Irish Research Council. Thank you so
And the
much.
farmers
Yeah.
whose
We
habitats
have a
we're
few
sampling.
questions. We have a few
Yeah.
questions for you, Nisha. So I would say keep your slide open and shared because you might be helpful to hop back and like look at a couple of things. I
Yeah.
try to grab like the in moment ones as much as possible, but
Mhm.
um Sinead said they're wondering if the nematodes found in raised bog would differ to those in a blanket bog, and if bog restoration would affect them, as in, are there less in re-wetted bog?
Yes. So, yes, the nematode families that were present in the raised bog were very much different from that ones in the fen peat or the woodlands or cutover scrubs. But the blanket bog, I have not done any blanket bog sampling, so I'm not sure about it. But yes, there are different families in different bog habitats.
Yeah.
And then coming to what was the other question? Yeah, in the re-wetted
So,
and the non-re-wetted. Yeah. So as I mentioned, like in only in this particular site, which is of improved fen peat grassland, there was a significant difference, but it would be very difficult for me to say at this stage because re-wetting or restoration, it kind of takes a long time for the ecology to kick in. So according to my observation, it says that the re-wetting reduces the pests. So, but to say it confirmly, I would like to, I would need more results, which I'm waiting on. So once I have that, I could say the answer for it very with a lot of confidence. Yes.
yeah.
Joshua asks, do we have nematodes living in us?
Yes, we do have nematodes living in us. So, just for example, the disease that is elephantiasis caused like it's like a swollen leg. It's caused because of a nematode. Yeah.
Interesting.
That's in
Interesting.
Ireland. Do we have any inside of us in Ireland? More
In
importantly,
uh, may not be, may not be. Or C.
Uh,
elegans, which is known to be a very model organism. It might be there in some kind of infections, but I'm not very sure about it. Yeah, if it's in
um,
Ireland or not. Yeah, yeah. Next.
we have another question, and I'm so sorry if I get anyone's names pronounced like very Americanly pronounced or just pronounced incorrectly. I'm so sorry. But Nasamu had asked, did you observe any correlation between the number and types of nematodes based on morphology? I'm so sorry.
morphology.
Morphology.
Yeah.
I'm trying. And amplicon sequencing. I hope that made sense. I'm so sorry.
Thank you so much for asking such a scientific based question. Yes, when we try to do the morphological and molecular kind of identification, it's very difficult for us to obtain 100% similarity in both the analysis because molecular is very in depth, but the sample size for molecular identification is very small. So when we are doing DNA extraction, it's literally from 250 milligrams of soil. So the situation arises that we are just using very little amount of soil as a representative of a big field, even though we are mixing it and doing all those kind of pre sampling methods. But it's in depth, but it's for very small amount of sample. But the morphological identification on the other side, it's a very promising kind of identification. But the problem with that is like it's very time taking. So the way I was telling you, like approximately 70 to 100 nematodes, that's what I take for per habitat, but that might not be the again overall representative. So it's a very tedious work. So we try to complement the analysis with each other, but to get 100% similar results, it's really difficult. Yeah.
Um, Another question, is there any research using nematodes to control scrub such as Sitka spruce or lodgepole pine regen alongside measures to raise the water table for peatland restoration? Accepting that this is potentially a risky endeavor, especially near forestry/agricultural sites.
Okay, there are a few publications, just one or two from Germany and from Slovakia, which have been using nematodes as indicators in the peatlands and to control their trying to compare the restoration efforts. So, but I'm not sure if it's particularly for Sitka spruce or any particular trees, but it's more kind of a general information of the nematodes using as indicators. So if you are interested, I can kind of share that information. I hope this answers your
Just,
question. Yeah.
Yeah,
and
Anusha,
again,
if you would share that with us, because we can put that in when we send out the recording to everybody too, so that way everybody
Yeah.
can kind of see that research as well.
Okay.
and
Perfect.
just to comment again, just where we have done rewetting, where there is spruce, generally the rewetting does for the Sitka spruce anyway. That's what we've observed.
Mhm.
So just as a footnote.
Um, Another question, were there any nematodes which were an indicator species of a natural bog?
Yes. So that's a very interesting question. Yes, there is one nematode species. I hope I have the picture straight away. Okay, so it belongs to the family Teratocephalidae, which means like it's like a monster-headed nematode. For me, it looks very beautiful because it has very typical characteristic features. Its cuticle is very much annulated. It's very beautiful, and these nematodes were found in very abundantly in the raised bogs. Yeah. So if I have that
Awesome.
picture, I might show it, but I do not know how quickly I can find that.
If you find it later too, we can
I'll
always, like I
share
said,
it.
include
Yeah,
it. Yeah.
yeah.
That's why we love a follow-up email. We can just kind of slide everything else in there. Awesome. Um, Another question, are there more nematodes in peatlands compared to other field soils?
No, field soils or peatland habitats, in terms of abundance, I might not be able to tell you exactly how abundant, but I think these nematodes are found in every environment. So recently, there is a study where people have found nematodes and are trying to study the nematode families that are present in Antarctica and such kind of extreme temperatures and weather conditions. So nematodes are present everywhere. So that's the reason why they can be used as bioindicators.
So
That's really
but I think is a question.
exciting.
I suspect the question, maybe somebody can confirm, but I think would the question be if we're looking at peatlands and maybe because we had peat grasslands compared to other field soils, maybe would that be mineral soils? But you're saying they're everywhere. But again, maybe would you be able
Yeah.
to
So
comment on the abundance, relative abundance?
yeah, so the relative abundance, some kind of families would be highly abundant in bogs or the health raised bogs, but then in the fen peat kind of alkaline type of environment, there will be different families present, which are relatively higher abundant than the other nematodes. But again, in the mineral soils, there will be different type of nematodes, like mostly omnivores or predators. The composition of the nematodes or the nematode communities changes, but the abundance could, so it will be like more, okay, 90% of the bogs will be having 90% that are belonging to moss feeders or the teratocephalidae kind of bacterial feeders, whereas the mineral soils might have more of omnivores that feed upon other nematodes or other organisms. So that's how it would be. There would be relative difference in the abundance, but not in the absolute abundance. That's what I meant. Yeah.
Awesome. Another question. So knowing the nematode pattern helps bog restoration efforts by choosing the right plants. Now, there are some heavily degraded bogs where nothing grows back at all. Can this research help finding out why this is?
Sorry, Kayla. Can you please repeat the question?
Of course, yeah, yeah. So they say, knowing the nematode pattern helps bog restoration efforts by choosing the right plants. Now, there are some heavily degraded bogs where nothing grows back at all. Can this research help with finding out why this is?
Okay, so when we're talking about the degraded bogs, yes, nothing much grows there, but we can still see the mosses growing there. So in
I
those
think
kind of,
just sorry to cut across the initial. I think maybe is it Andre? He might be referring to where like you have the board and ammonia bogs. I suspect he's talking about these kind of brown deserts where it's bare peat, encrusted,
Um,
exposed. Yeah.
yeah, so there are some bogs in the Wicklows, for instance, that have the hilltop basically has a huge erosion problem. There were big fires 40 or 50 years ago, and ever since nothing is growing back.
Mm,
There's the fungi isn't right in the
Hmm.
bog in the soil, and everything else seems to be off. And now they start planting heather, which is a
okay,
Hmm.
plant that would come very late in the evolution of a bog. But so that's a hit and miss again. So would this research maybe help finding out whether that's actually promising approach based on trying to plant something that hasn't grown in like 40 or 50 years?
okay, that's actually a very interesting question. So yes, so for that, we need to do some kind of research on those soils. And yes, if we investigate what type of nematodes are present in there, and if we say like there are more of the bacterial feeder ones, then yes, based on that information, we can like select some certain kind of plants that could grow in those lands. Yes, that could give us an indication of what plants can be chosen to be grown in those fields. So yeah, so if there is a dedicated research lab for nematode study in SETU, so we can help you in selecting the crops and selecting the plants that can grow in these lands. Yeah,
that would be awesome. Yeah, I will be in touch. Thank you.
Thank you.
That's so interesting. Another question. If nematodes create a cyst, so thinking like in the human body, if nematodes create a cyst, is there any connection of cancer in nematodes in us?
As far as my knowledge is concerned, there's no literature or there's no evidence so far which says that the plant cysts have any relation with that of the human cysts. But these nematodes evidently cause cysts or galls in the plants. But I'm not sure if that is anywhere related to the human cysts. No.
That's an exciting thought. Like that seems like a very exciting sort of extension of research to sort of understand that.
Yeah.
I think it's easy to be a human and not think of ourselves as also being a biological organism, you know?
So that's like
a
research
humbling.
on its own. It's a research proposal on its own. It's like a very interesting question to find out. Yeah.
Yeah.
Awesome. I'm just looking for any other questions we have.
Yeah.
Any of our
There was a follow-up there. There was a follow-up there, Kayla.
Oh, there was?
Just from Jack. Yeah.
Let me see. Oh, yes. Douglas, I agree, but sometimes we cannot raise the water table depending on the erosive feature. For example, severe gulling, and in those areas, the regen can be, I hope I pronounced that. Sorry.
Regeneration.
Can be quite severe.
Yeah. So I think that I'm assuming, Jack, I don't know if you wanted to clarify that that's probably sloped blanket bogs in the mountains, and then obviously we're doing it in the raised bogs, so it's nice and flat. It's very easy to raise the water table, but on a slope, water table isn't going to do that, and the regen in this case would be the spruce just seeding into the bog and taking over. So yeah. So then I suppose the interest, Jack, again, if you want to clarify and if I'm getting it wrong, I'm presuming he's maybe getting at nematodes that could actually control that, because for the restoration efforts on those peatlands with the spruce and lodgepole pine coming in, this would be a big problem.
Definitely. Interesting. Okay. And then this is just sort of an added comment from Neil Warnock. Someone had asked.
Sorry, Kelly. So then I don't know if you'd be able to comment on that, Anusha. I know you love your nematodes. You're talking about the beauty of them. I think we need to acquire that taste yet. Perhaps we need another few presentations. We're not quite there yet. So I think, you can use nematodes to control lots of things. So do you think that could be something
that should be something to look into.
Be looked at. So in terms of spruce and lodgepole pine, there may be nematodes that can actually be used or identified to stop those infestations where they're trying to restore the bog.
Yes, definitely, yes. So it would be something for future research. So.
Awesome. And then I'm just looking at the other questions here. So this is just a sort of just an in-chat follow-up. I don't know if there's any comment you want to have on that, but Neil had responded to nematodes living in us. Generally, no, the vast majority of us do not have nematodes living in us. Children can get pinworm, but it is easily treated. You can also get ascaris or toxicara. Hoping I'm saying that right, but both are rare. Gosh, you guys are putting me through my paces today. Trichinella, thank you. Is an infection you can get from the uncooked pork, which could be dangerous, but again, quite rare since most aren't eating handfuls of raw pork. I would recommend not doing that. That's my non-scientific but professional recommendation. Okay. And then any of our freshwater source from bogland transmit to humans and other plant life. So I'm not sure if that's in relation to nematodes being maybe found in water.
Yeah, they are found in the water, but if they're transmitted to plants, they are definitely transmitted to plants, but humans, I think it still needs to be investigated. I'm not sure. I haven't found any human parasites so far in the peatlands, so I'm not sure about it.
And then let me just see here. Hold on, my whole view changed. There's my chat. There we go. Um, I just want to give us something to look at as well. And I think trying to see if there's anything else. We're gonna find today's super interesting slides. Andre, we'll be sending those out along with the follow-up information. So if you're registered for the webinar, we have an email list, so we'll be sending all that out to you. And then I need to see question. I also work on from Jack. I also work on quite a few intermediate bogs, so a mixture of blanket bogs and raised bogs.
And
Interesting.
he also said his mic isn't working, so he wasn't able to
Okay.
clarify himself. But yeah, so it's not just raised bog, but blanket and raised. This is for the control of the pine or the spruce
Got it.
on the damaged bog. Yeah.
And then Andre, I think this is the last one. Andre says, is there a GIS nematode map for
For Ireland.
Ireland?
Not as such. So the team in ICTU is again trying to develop some kind of database for it. So if I have anything updated information about it, I would like to share in future, but not anything as of now.
Very exciting. Awesome. Well, I just want to take a moment and thank Dr. Anusha for leading us through this really exciting webinar. I have the unique perspective of someone who's not a scientist, and I just find this really, really fascinating. This is just such an, I'm so sorry to hear my dog whining. Um, he is also very excited about nematodes. Um, But this is just really exciting research, and learning from Anusha has been just an absolute thrill, and it's helped me learn so much more about also just like the environment around me. And it's just been great. So thank you so much for presenting. We're so grateful to have you. And we're excited for you. We're
Before
excited for your
people leave,
future.
Kayla.
Yes. Yep. So I just wanted to tell everyone that we are actually up for an award. Give me one second. There's someone in chat. Perfect. So Greenwich Church in Ireland is actually up for an ARIA award. I could talk about this all day, but we need your vote. So let me, if you actually scan the QR code, there is a link to where you can vote. Um, and we were up for the Agricultural Research Inspiration Award. And we're one of two Ireland organizations that are finalists, and it's very exciting. And we wouldn't have been able to have even gotten here if it wasn't for our farmers, if it wasn't for the research being done, obviously by folks like Dr. Anusha and also Dr. Doug McMillan, all the doctors. And so it's very exciting. You can read more about it through the link. But we would love for you to go vote for Farm Carbon EIP.
Yeah. And
And
this is a EU European-wide competition.
yep,
So, that's it. It's public vote is very important. So if you can take 30 seconds, as Kayla was saying, to scan that and vote for us, that would be brilliant and help us
yep,
with
you
follow
should
the
see
work.
it. It should be the first link that's on the link tree. So we're really excited. We'll put a link to it as well in the email, but we're very excited. A couple of our farmers are actually going to fly out for the award, which is great. And we're just so thrilled that we can share this with them because we wouldn't be able to have done this without them. Period. Full stop. So, but I think that's about it, everyone. As I mentioned, I will be sending out an email with the recording with different information, some of the information that Anusha had mentioned that we'll share out with you. But also feel free to send us any questions as well in response to that email. We're very grassroots, so you'll get a human being. It'll be probably me or Doug. uh, but thank you all so much for watching, and
Just want to
Thank
say
hopefully we'll
you,
see
one
you in the
thing,
everyone.
next
Kayla.
one. Yes.
Sorry, just want to say one thing. So I try to answer the questions as per my knowledge. So if there are any other questions from anyone, if they can email me,
Mm.
I'll be happy to answer them.
Yep, and
So
we'll have Anusha's email in there, so
yeah,
you should be able to also send an email her way directly, which is exciting.
thank you.
Awesome. Well, thank you so much for joining. Have a great day or great night or wherever you are in the world.
Thanks, everyone.
Bye.